Leukocytes are the cells of the immune system. Leukocytes are motile, flexible, true cells found in tissues. They contain a nucleus and organelles but lack hemoglobin and serve to help defend the body against any kind of pathogen. They have unique movement abilities such as diapedesis, which is the process of leukocytes entering tissues by act of squeezing between the small endothelial cells of the blood vessel's wall. Chemotaxis is another process by which leukocytes can attract each other to sites of infection due to the release of these chemical signals via dead cells or damaged cells or even pathogen. Another movement is called margination, which is the form of transient bonds between the blood vessels using adhesion receptor. On the left here, you can see the distribution of different kinds of white blood cells. We have 3 granulocytes, neutrophil, eosinophil, basophil, and we have the 2 agranulocytes, the lymphocytes, and the monocytes. And you can see the distribution of them inside your blood. Let's start with the first type, neutrophils. Neutrophils are the most abundant white blood cells. They're named for the neutral, pale-colored granules inside a light, lilac-colored cytosol. They're characterized by a unique multilobular nucleus. They could even have up to about 5 lobes, and this is why we call them polymorphonuclear. Generally, they remain in circulation about 10-12 hours, and when they enter in tissue spaces, they phagocytize pathogens, release enzymes, and they can kill their target. Increased neutrophils oftentimes indicate an acute bacterial infection inside the body. The second type is called eosinophils. Eosinophils are large bilobe nuclei. They get stained with bright red acidic dye called eosin. As a result, their granules are more of a reddish pink-orange color. They are also able to phagocytize compounds, but they target antigen-antibody complexes and also allergens. So if the body gets infected by a parasitic worm, the eosinophils release chemicals that will mediate the worms. The third class is called basophils. Basophils are the least numerous of the granulocytes, making only about 0.5 to 1%. They exhibit many deep blue violet granules when they're stained, and they have a bilobular nucleus. The basophils can release histamine, a capillary dilator, and also heparin, which is an anticoagulant, which is released during inflammation and also allergic responses. They may also contribute to allergies and anaphylaxis. Let's take a look at the agranulocytes. We have the lymphocytes and the monocytes. The lymphocytes reside inside the lymphatic organ tissue. They're the size of about an erythrocyte, making about 20 to 40% of all leukocytes in the blood. They have this dark staining nucleus and typically very round and small. The cytosols appear more of a light blue color. And when they activate, they swell up and grow larger. There are 3 subcategories of lymphocytes. There are T cells, T lymphocytes. They manage direct immune responses by directly attacking foreign cells and virus-infected cells. We have B cells, B lymphocytes, that become plasma cells and are able to produce antibodies. And we also have another one called NK cells. NK cell stands for natural killer, and these cells are responsible for attacking abnormal infected cells and oftentimes regulate cancer cells. The last type is called monocytes. These ones can be up to 3 times the diameter of an erythrocyte and make up about 2 to 8% of all the white blood cells. Their hallmark structure is a kidney-shaped or C-shaped nucleus, and after 3 days in circulation, these monocytes will exit the bloodstream and take residence within tissues, where they can transform into large phagocytic cells called macrophages. These macrophages can then engulf bacteria, cell fragments, and debris. Focus on disease. Infectious mononucleosis. What is this? This is called the kissing disease, and this is caused by the EBV virus, the Epstein-Barr virus. This virus is transmitted through saliva, semen, and human blood. As a result of this, it can cause flu-like symptoms. The virus will cause the T cells to swell up and enlarge as they fight against infected cells, making them resemble monocytes, hence the name mononucleosis. There is no treatment available, but your body will depend on the immune system to fight it off after a few weeks. Blood type and transfusions, an intersection of red and white blood cells. Blood type with the ABO system and the Rhesus factor, RH factor. So surface antigens or agglutinogens are molecules located on the plasma membrane of erythrocytes or red blood cells. These are best known as antigens that code for the ABO blood group. The presence of an A antigen and the B antigen determines your ABO blood type. So for example, type A blood has the A antigen. Type B blood will have the B antigen. Type AB blood will have both antigens, and type A blood would have no antigen. So as you can see on the image, the circle represents the A antigen, and then the green square represents the B antigen. So the ABO surface antigens are accompanied by specific antibodies in the blood. These antibodies, also known as agglutinins, are these Y-shaped proteins in the plasma that will recognize these antigens and perceive them as foreign. So in the ABO blood groups, you're going to also contain the anti-A and anti-B antibodies. A person that has type A blood is going to have the anti-B antibody. A person that has type B blood will have type anti-A antibody. A person with group A and B antigen will have no antibodies, and the person that has type O blood will have both kinds of antibodies. Now, the Rh is an indication of the rhesus factor, which is another antigen found on the cell membrane. If it's positive, we say that it's positive for the rhesus factor, and if it's negative, we say that it's absent for the rhesus factor. So the Rh blood type is determined by the presence of the Rh antigen. In some texts, we call that the D antigen. So if the D antigen is present, it's going to be positive blood type. If the D antigen is absent, we call it the negative blood type. So identifying the blood type using antibodies. So combining these incompatible blood types can cause antibodies of one type to bind to the surface antigen of the second type. As a result, there's a reaction called agglutination, clumping of the blood cells or blood clot. And so you can see these dark specks over here representing the clumping and clotting effect when you appropriately add the antibody that matches with the antigen. As a result, hemolysis occurs, and this is a process where the blood cells begin to rupture due to that clumping. If this happened in a patient, this will cause organ damage or hemolytic reaction. So because agglutination and then leading to hemolysis, only certain blood types are going to be compatible for certain kinds of people. For example, type O is considered the universal donor, right here. And then type AB positive will be considered the universal acceptor, because they can receive anybody's blood. So let's take a look. So if you're a recipient that has type A blood, that means you're going to have the A antigen on your red blood cells, which means you're going to have the B antibody, therefore you can accept both A and O. If you're type B blood, you're going to have the B antigen on the cell, and you're going to have the type A antibody, and then you're going to be able to accept both B and O. If you are AB blood type, you'll have both antigens of A and B, and no antibodies circulating inside the plasma, but you can accept A, B, and O. And the last but not the least is an O blood type will have no antigens, but both antibodies, and that means you can only take blood from O. So just remember, if you are a positive blood type, you could take blood from a positive and a negative, but if you are a negative, you should only take from the negative. So AB is considered the universal recipient, and then the O blood type is the universal donor. So one clinical application is what we call the Rh rhesus incompatibility in pregnancy. During pregnancy, if a mother's blood type is an Rh negative and her fetus blood is an Rh positive, there can possibly be a medical complication. Generally, in the second pregnancy, not the first one, the mother could be exposed to the Rh antigen during birth because it is a bloody process, and then produce antibodies against that Rh protein. During the second pregnancy, if the fetus is again Rh positive, then the Rh antibodies could potentially cross through the placenta, resulting in hemolytic disease of the newborn baby, leading into a condition called erythroblastosis fetalis. And erythroblastosis fetalis occurs in the baby and presents with certain kinds of symptoms like anemia, hyperbilirubinemia, and may even develop heart failure and must be given a transfusion. So this is a visual showing you what happens with the maternal tissue, fetal tissue. Mother blood is negative blood type, and then the baby's blood is positive blood type. Because of the mixing of the blood exposure, the mom's blood system will then recognize the Rh rhesus factor and begin producing antibodies for the second pregnancy. Then if the second infant is also Rh positive, there's a chance that the antibodies could cross over between the maternal tissue to the fetal tissue and cause hemolytic disease.